Okay, the subject of today's lecture is enzymes as drug targets. And what I'll be covering over the next 40 to 50 minutes is really an introduction to how we can consider enzymes as drug targets, the different ways in which we can target them, and I'll give you some examples of how this is done in the real world. So examples of drugs that directly target enzymes and which have demonstrable clinical benefit. Actually, some of these are from many, many years ago and are still in clinical use, which gives you an idea as to how important enzymes are. In terms of lecture objectives or learning outcomes, at the end of this lecture and when it comes through to exam time, you should be able to discuss how enzymes can be drug targets and give examples of inhibitors that are actually used in the clinic as drugs. You should be able to define the kinetic parameters of enzyme-catalyzed reactions, in particular Km and Vmax. You should be familiar with these from part one, but I will briefly touch on these again as a reminder. And also the effect of different types of enzyme inhibition and inhibitors on these characteristics of enzymes. So one of the ways in which we define targeting enzymes as drug targets is by the way in which their kinetics are altered by inhibition. And also explain the meaning of an inhibition constant as a measure of the potency of an enzyme inhibitor. So as I said, what I'll be going through is a very brief introduction to enzymes. This is really just a recap. I'll be talking briefly about enzyme kinetics. Again, this should be building on knowledge that you had from part one. For both SPS and pharmacy students, if you're not clear about how to think about enzymes in terms of their kinetics, I would really, really strongly recommend going back to your part one notes or using the wide variety of resources that there are on the internet, which will help you to understand enzyme kinetics. Because really that's at the heart of understanding how we treat enzymes as drug targets. And as I said, I'll go on to talk a little bit about examples of enzyme drug targets and drug mechanisms of action. So the opening part of this is really thinking a little bit about enzymes and why they're important. So enzymes are biological catalysts. What they do is they either accelerate or increase the chances of a particular chemical reaction occurring. This is really defined by their three-dimensional structure. Essentially, they're protein machines that carry out tasks within the cell. And this is a myriad of tasks. So enzymes are at the heart of almost all of the cellular pathways that you think about in terms of cell biology and also important for clinical medicine. They are central to all aspects of cellular function, and that includes both bacterial and eukaryotic. And one of the examples that I'll give you later on in this talk is an example that highlights the importance of the distinction between bacterial enzymes and eukaryotic, and in particular, mammalian and human enzymes. Why are they important? So in terms of drug discovery, in terms of pharmacological treatment of disorders, they are the single biggest group of drug targets in the human body. And they account for, it's actually a little bit more than this now, up to 47, nearly up to half of all of the drug targets that are listed as being clinically targeted by drugs in clinical practice are targeting enzymes. So if you're taking a drug for any kind of condition, the odds are about 1 in 2 that this will be targeting some kind of enzyme. That runs all the way through from ibuprofen to advanced drugs which are in use in clinical oncology. And again, I'll give you examples of a couple of these later on. So clearly, from the point of view of understanding drug discovery, targeting human disorders, understanding enzymes as drug targets is critical. Without an understanding of that, actually you don't understand half of the drugs which are in clinical use. And what do enzymes actually do? So enzymes are proteins. They're three-dimensional organizations of amino acid polypeptides, often with cofactors, and they have specialized binding sites, pockets on their structure that are specific for certain substrates. Once bound, a substrate is modified. So here we have a cartoon of a communal garden enzyme with a specifically shaped active site. This is determined by the three-dimensional fold of the protein. That is specific for a particular substrate. Again, this is a cartoon exemplifying how this occurs. The substrate binds to the active site, And the enzyme changes shape slightly as the substrate binds. So this is a very dynamic process. And actually, in terms of understanding the kinetics of all of this, remembering that this is a dynamic process, not a static process where something occurs and then nothing changes. Proteins are dynamic assemblies within the cell. So as the enzyme changes shape, there is an interaction with the substrate, often using ions to directly target some of the chemical bonds in that substrate, modifying the substrate and resulting in the release of products. So essentially you have a several-stage process. You have the substrate entering the active site of the enzyme. You have the formation of an enzyme-substrate complex, then the enzyme products complex, and eventually the products leave the active site, freeing up the active site for new substrates to bind. You can represent this energetically, same as you would represent any catalyzed reaction. So catalysts can be inorganic as well as organic. You can have metals that catalyze reactions. Any of you who have done any chemistry in the last couple of years will be very, very well aware of that. And so what an enzyme does is it basically alters the energetics of a reaction. So here we have a simple projection of what a simple chemical reaction looks like. And with the vast majority of chemical reactions, you have a hump to get over, an activation energy, which is required for the reactants to come together and react and change into products. In this case, we've used the example of carbon dioxide and water. Now, normally, this activation energy prevents that reaction occurring. So, for example, if you have a carbon dioxide mixed with water, the majority of that carbon dioxide will not react with the water to form a different chemical. And so this activation energy basically prevents the reaction occurring. What catalysts do, and this can be a metal catalyst or an enzyme, is it reduces that energy of activation. So it makes it much more likely that the reaction will occur and that the products are produced. And this is basically what enzymes do within the cell. The key thing to note about enzymes is their specificity. They have evolved through millions and millions and millions of years to be as specific as possible for an individual reaction. So they will bind a specific product, a specific substrate, and they will produce a specific product. We can represent this in terms of enzyme kinetics by looking at the reaction rate plotted against substrate concentration. The idea being that as you increase the concentration of a substrate, the likelihood of that substrate binding into the active site and being processed increases. The higher the substrate concentration, the more likely it is that reaction will occur, and the reaction rate increases in response to that. And there are several key definitions which you should be familiar with from part one, but again, I would really emphasize that it's important to really understand what these define. The key considerations here are Vmax, and this is the maximum rate at which an enzyme can carry out a reaction. So given a projected almost infinite concentration of substrate, this is the maximal velocity that a reaction will proceed in the presence of an enzyme. The other key definition to think about is the Km, and this is the substrate concentration at which the enzyme will work at half of its maximal velocity. And you can see what's been plotted here is a Vmax running along the top here. And when you go down to half of Vmax, the substrate concentration at which this is achieved is the Km, and it's done by moving across to the x-axis here. Now, there are lots of examples of enzyme kinetics in practical labs. The pharmacists among you will remember that in part one we carry out a very simple enzyme kinetic analysis in the laboratory looking at the activity of lactase. Lactase basically is the enzyme that we use to break down lactose, the sugar found in milk, break it into D-galactose and D-glucose. Without lactase to break apart the bond between these two sugars, you cannot generate energy from lactose. And so actually milk is no good to you if you don't have a functioning lactase enzyme. What we carried out in the practical last year was using an artificial substrate called OMPG, which is hydrolyzed by lactase, to produce a coloured product from the reaction, and this is shown down here. For those of you in SBS, I actually have a video of this, which I will be posting onto the website, so if you're interested in learning a bit more about this practical, there's now a JOE video, Journal of Visualized Experiments video, which you can look at to get a bit more understanding of this. So these are enzyme reactions that we can measure in the laboratory, we can quantify them, and we can calculate those key parameters, the Vmax and the Km. And what we would do is we would purify the enzyme of interest and carry out a series of reactions at different substrate concentrations, measure the rate at which the reaction occurs. With lactase, what we can do is we can measure the production of orthonitrophenol, which has this particular yellow color, so we can actually measure this in a colorimetric fashion, and we're then able to calculate the Km and the Vmax. This is most easily done by actually plotting the data in a slightly different way, plotting it on a Lineweaver-Burke plot. What this does is it takes a reciprocal of the velocity and substrate concentration and plots those, and what you get is a linear presentation of the enzymatic kinetic data. The reason this is useful is that it allows you to calculate the Vmax and the Km at points at which it intersects the x-axis and the y-axis respectively. What I'll be doing over the next few slides is actually demonstrating how we can use this type of information to understand how an enzyme is being modified by a specific inhibitor. So there are two key classes of enzyme inhibitor that you need to be aware of and need to understand. I'll talk a little bit about a third class towards the end of this lecture. However, the third class is much less common. These are competitive and non-competitive inhibitors. And actually the difference between them is really quite simple. The first, which we've got shown here, is the simple reaction. So you have, again, this is a cartoon representation of an enzymatic reaction. You have the substrate binding to the enzyme. That substrate enzyme complex then undergoes a conformational change. The protein changes shape slightly. This puts stresses on the internal bonds of the substrate, which results in a reaction. Very, very frequently, for the examples I'll be giving you, a hydrolytic reaction, which requires water. Now, if you inhibit that reaction, there are several ways in which you can do it. One is by competitive inhibition. So there you have an inhibitor, a chemical, which has a very, very similar shape, three-dimensional organization, to the substrate. Basically what it does is it fools the enzyme into thinking that it is the substrate rather than the real substrate. So what happens is you have this interacts with the enzyme, has the same shape, and again this is a cartoon representation, but if you try and have the concept of this in 3 dimensions in your mind, this really is at the heart of how we target enzymes. So you have an inhibitor which shares some of the properties of the substrate, but importantly is not the substrate and would not be processed in the same way. It wouldn't be broken apart into products that would allow the enzyme to carry out its normal role. Now, if it's a competitive inhibitor, this will basically exchange backwards and forwards with the substrate. So the greater the concentration of the inhibitor as a competitive inhibitor, the more likely it is, just by chance, that the inhibitor will bind to the active site rather than the substrate. And so it's directly competing against the substrate for occupancy and binding of that active site. And that is a fairly simple concept, really. In terms of non-competitive inhibition, exactly the same kind of reaction is occurring, that you have a substrate which is of a similar shape to an inhibitor which is of a similar shape to the substrate, but instead of binding and then being released and being engaging in a dynamic exchange process with the enzyme in question, actually what occurs is a covalent modification of the substrate of the enzyme. So what you can see here is a representation of the way in which we can represent this in terms of enzyme kinetics. And really, this is just another way of showing exactly what I've shown here in this cartoon. So here we have the enzyme and the substrate. The enzyme binds the substrate and forms an enzyme-substrate complex. You then have the enzyme releasing the products. What we're doing here is really representing that with letters rather than images. So here you have the enzyme and substrate, you have the enzyme-substrate complex, and the enzyme and product. And these are all in a very dynamic process. If you have inhibitors, you can then drop those in to bind to the enzyme and the inhibitor, basically blocking occupancy of the enzyme active site and preventing the substrate ever even binding. And this gives you one inhibitor constant to consider. A second type of inhibitor will bind to the enzyme-substrate complex, and so will then form a complex with the enzyme, the substrate, and the inhibitor, giving rise to an alternative inhibitory constant. And you can represent this using the same Lineweaver-Burke plots and actually get an idea from the enzyme kinetics as to what is happening. So here's the same plot that I showed you before. This is the reciprocal of the substrate concentration over the velocity. Here we have the point at which the best fit line crosses over the y-axis, and this gives you 1 over the Vmax. Here on the x-axis we have negative of 1 over the Km. Now, the way in which an enzyme reaction will respond is very different depending upon what type of inhibitor the enzyme reaction is exposed to. So if you have a competitive reaction, what you have with increasing concentrations of the inhibitor, you have an increasingly steep line formed in a Lineweaver-Burke plot. The important thing to note here is that what isn't altered is the Vmax. You can see that the point at which this plot crosses over the y-axis is exactly the same. So you're not altering the total velocity of the reaction. If you had enough substrate dropped into the reaction, it would still proceed at close to the Vmax. What you are altering is the Km. So you can see that the line is altered so that the Km is actually altered. And this allows you to determine experimentally whether or not you have a competitive or a non-competitive inhibitor. A mixed type of inhibitor I won't go into in much detail. This basically has properties of a competitive and a non-competitive inhibitor. In the context of this lecture, the important one to focus on here is the competitive and non-competitive inhibitors. So with the non-competitive inhibitor, what's happening is that the inhibitor is binding to the enzyme, and it covalently modifies, or basically it cannot be released by the enzyme as would normally happen to the substrate and the products. So what happens is that by binding to the enzyme, you're basically taking that enzyme molecule completely out of the enzymatic reaction. It is no longer able ever to break down, bind a substrate and result in the production of its products. And so here, what you get actually is a gradual increase in gradual decrease in the Vmax, as shown here with 1 over Vmax. So as that increases, actually the Vmax decreases. And what this represents is individual enzymes being removed from the pool of enzymes capable of carrying out that reaction. By covalently modifying the active site, you prevent that enzyme ever binding the substrate ever again, and that removes it completely from the possibility of carrying out the reaction. And one of the probably one of the most familiar types of drug used to target enzymes does exactly this, and I'll talk about it in a few slides' time. So one of the key things to think about, and here I'll start talking about some of the examples of enzyme inhibitors that are used in clinical practice. And the example that I'll start with are enzymes targeting infectious agents, most notably bacteria and viruses. So if an enzyme produced by an infectious agent can be switched off, inhibited in some way, shape or form, this can be fatal for the bacteria or virus in question. And if you're trying to prevent a viral infection or a bacterial infection, then clearly having a bactericidal or virucidal impact is a very good outcome. This is particularly the case if a bactericidal drug, a drug which actually kills the bacterium, targets enzymes which are only found in that bacteria and are not found in the human body or in the human proteome. And if you think about this, actually that makes a lot of sense. If you have a human enzyme that matches up to the bacterial enzyme, when you treat an individual with that drug, yes, you'll kill the bacteria, but you'll also be killing human cells as well. And so in this context, and particularly in terms of targeting infectious agents, the ideal enzymatic drug target is one which is found in a bacterial proteome or a viral proteome, but is not found in the human or animal which is being treated for that disease. And again, for viral enzymes, this becomes a little more complicated because of the different status of viruses as independently replicating agents. Obviously, they require many of the components of a host cell in order to replicate. So this is a slightly more complicated problem. And most of the antivirals that are present in the market and are able to be used to treat viral infections target viral replication, basically identifying enzymes which are found within a viral proteome and which are not present in human cells. So if a cell is infected by a virus, that particular enzyme will be present in the human cell, but it will not be found in cells that are uninfected by the virus. So the first example that I'll provide to you is the example of DD transpeptidases. These are a class of enzyme that are found in bacteria and carry out cross-linking of the peptidoglycan wall subunits for bacteria. These are also known as penicillin binding proteins, as will become very, very clear in the following slides. So essentially what these do is that they bind to the peptidoglycan chains, and they carry out a cross-linking reaction, allowing the bacterium to form a strong bacterial cell wall. Clearly, a bacterial cell wall is something that isn't found in human cells. We have the cell membrane. We don't have that capsule-like bacterial wall on the outside. And so this is a reaction which is unique to bacteria infecting human cells in the context of clinical medicine. So the transpeptidase binds to the peptidoglycan wall, forms a cross-link in an enzymatic reaction. So here you have the substrates binding, cross-linking occurs, and the enzyme goes off and does it all again. Now, this process is very, very important for bacterial replication. If a bacteria wants to replicate, it needs to build more cell wall in order to undergo binary fission and generate progeny. So if this process is in any way blocked or altered, this has a very, very serious impact on bacteria's ability to replicate. And this is basically what penicillin targets. So penicillin is a beta-lactam antibiotic, and it binds to the active site of the DD transpeptidases, the penicillin binding proteins, and acts as a non-competitive inhibitor. So what you do is you treat the bacterium with a beta-lactam inhibitor. Here's a cell wall, here's the bacterium inside with its cell membrane. By inhibiting the cross-linking of the bacterial cell wall, you prevent the formation of new cell walls, and eventually the bacterium continues to replicate inside in terms of its cellular contents and will bud out, form a spheroplast, and that is destroyed because it is no longer protected from the environment and will rupture and lyse, killing the bacterium. So this is a bactericidal approach to treating bacterial infections. And essentially what beta-lactams do is the beta-lactam structure goes into the active site of the DD transpeptidase, and this is a crystal structure, three-dimensional representation of the DD transpeptidase with a beta-lactam stuck in the active site. And what occurs is that these form the beta-lactam ring, which is shown here, is energetically a very stressed structure within the chemical structure of beta-lactams. This breaks open and forms a covalent bond with the active site of the DD transpeptidase. If you've got that covalently bound into the active site, there's no way that peptidoglycan is going to be able to bind the active site, and so you have a non-competitive inhibition of that enzyme's ability to work. Now, of course, bacteria have fought back up against this, and over the course of millions of years have developed their own approach to trying to get rid of these beta-lactams. And the way in which they do this is by making their own proteins to counter the impact of beta-lactams. And they produce beta-lactamases, which bind to the beta-lactam structure and hydrolyze that ring in that beta-lactam structure in a controlled way from the bacteria's perspective. And this actually is what underpins, in terms of penicillin resistance or methicillin resistance, this is what underpins the drug resistance that we see to the penicillin class of antibiotics. So when you hear news about methicillin-resistant Staphylococcus aureus, what's driving that is the expression of an enzyme. It's being driven by the expression of a beta-lactamase, which is able to bind to that chemical structure and prevent it from targeting the DD transpeptidases. So in a way, it's the bacteria are carrying out a sort of enzymatic chemical warfare. We, of course, can fight back against that, and what the different classes of penicillins do is these structures have been modified to prevent them being hydrolyzed by the beta-lactamases. And this then means that they can target the DD transpeptidases. The bacteria have evolved to counter that, developing new beta-lactamases, which combine the new generation of beta-lactam antibiotics and get rid of those, and so on and so forth. But this is really what underpins a key aspect of what is actually emerging as a public health crisis. Over the past 50, 60 years, we've been able to treat bacterial infections, such as infections from Staph aureus, with really almost no effort because of the beta-lactams. As the bacteria's enzymes have evolved, that has actually prevented us treating disorders which have almost been eliminated as a major source of mortality in the developed world, and where antibiotics like this are available. So this really exemplifies exactly how important it is to understand enzymes as drug targets, and actually really understand it in quite a lot of depth, because the underlying protein biology of this is very important. What I've shown here actually is a beta-lactamase from Staphylococcus aureus. So this is the crystal structure of the enzyme that the bacteria make to fight back against the antibiotics that almost all of you will have been exposed to at one or other point in your life. The next example I'll give you is an example of an antiviral. And the example I'll give you is a drug that targets human immunodeficiency virus protease. So the replication of HIV within cells and its ability to infect human cells is very much dependent on a set of enzymes that the virus produces when it infects a cell. And there are actually several of them which are particularly critical in terms of targeting HIV. The one I'll talk about today is the HIV protease. So this is a representation, or a cartoon representation of the HIV virus, and inside the capsule you have a reverse transcriptase, but you also have a protease, which allows the processing of the proteins that are produced by HIV when it infects cells. So HIV infects a human cell, it reverse transcribes its RNA into DNA. This is then inserted into the host cell DNA, and it becomes part of that cell. But it requires a protease, which is encoded in its own genome, to produce new viral capsids. And so this has been targeted for many, many years now as part of the effort to target HIV and to suppress replication of the HIV virus, thereby preventing individuals developing acquired immune deficiency syndrome or AIDS. And this is an example of a drug which does that. This is tazanavir, which is produced by Novartis. And what this does is it jumps into the active site of the protease. Proteases are a class of enzyme which bind other proteins and break them into separate pieces. So exactly the same concept of that enzyme-substrate reaction. Here, the enzyme is a protease and the substrate is a protein. The protease binds the protein, in this case an HIV protein produced from within the HIV genome, and it breaks it into several sections which then are allowed to go on and form the viral capsid, which allows the virus to replicate, let those the viral particles go off and infect other cells. So it really is a key part of the life cycle of the HIV virus. And what this drug does is it goes into the active site of the HIV protease, and it acts as an inhibitor of that protease, and it acts as a competitive inhibitor, preventing that protease working. If the HIV protease isn't working, then the HIV-infected cell cannot produce new copies of the virus, and thereby suppresses the infection ongoing in the individual infected with HIV. Going back to what I was talking about in terms of targeting enzymes which are not present in the human proteome, the HIV protease is actually structurally very distinct to the proteases which are found in the human genome or human proteome. And so an inhibitor that targets the HIV protease will not inhibit the human proteases which are essential for the function of all of our cells. If you were to inhibit proteases within your own cells, that would be catastrophic for your cells and would result in significant cytotoxicity, a lot of cell death, and a lot of cells being killed, and therefore causing real problems for the individual being treated with that drug. So again, highlighting the importance of the specificity of these reactions. It should also be noted that the other enzyme, one of the other enzymes which is targeted in HIV therapies is reverse transcriptase. Our genomes do not have a reverse transcriptase, an enzyme which is capable of converting RNA back into DNA. We have enzymes which transcribe DNA into RNA, but not the other way round. And so many of the other inhibitors which are used to treat HIV infection target reverse transcriptase exactly because of that, because it is an enzyme which is not present in the human proteome. The next examples that I'll provide are a little more complex because they're actually targeting human enzymes. So these are enzymes which are normally found within your cells and regulate physiological processes in your body to ensure that your body continues in normal function. And again, it's important to note, enzymes are key in regulating every single human physiological function. Whether you look at blood pressure, whether you look at DNA replication, enzymes are there carrying out tasks, catalyzing reactions, and allowing your cells to function. If your enzymes aren't working, these can cause very, very severe disorders. And actually, many inherited disorders are underpinned by mutations in enzymes which reduce or alter their activity. Now, by understanding that and understanding what a particular enzyme does in terms of a physiological reaction, you can target that enzyme function as a way of modulating human physiology. And in particular, if there is an underlying alteration in physiology which is resulting in pathophysiology, or the derivation of a disease state. And as I noted, changes in protein sequence caused by mutations in your genome can alter protein structure, alter enzymatic activity, and lead to human diseases due to altered enzymatic function. The protein that I spend most of my research looking at is a kinase, and I'll talk a little bit about kinase in a few slides, called leucine-rich repeat kinase 2. If you have a mutation in this gene, you develop Parkinson's disease. And you can tell that from birth. You can sequence someone's genome, identify that mutation at birth, and predict that that person will develop Parkinson's disease within a range of about 5 to 10 years. So this gives you an insight into what happens if an enzyme reaction is disrupted, in this case by a mutation. So an example that I'll give you of an enzyme, a physiologically important enzyme in human biology and in human medicine, is angiotensin converting enzyme. This is a key enzyme involved in regulating the function of the kidney. And again, if you think back to your Part 1 lectures about kidney physiology, ACE, or angiotensin converting enzyme, is one of the key enzymes regulating how your kidneys function. It's a protease, so going back to the example of the HIV protease, it's an enzyme that binds to other proteins, cuts them, breaks them into products, and those products go off and do slightly different tasks to the substrate. What angiotensin converting enzyme does is it binds to angiotensin 1 and cuts it. It acts as a molecular scissor, breaks it into two parts, and releases angiotensin 2, which is the active form of this peptide. This has a number of downstream consequences, not least of which is an increase of water retention. And so if you have ACE converting angiotensin 1 into angiotensin 2, this results in an increased retention of water, so decreased excretion of water in the form of urine. And that, of course, can increase the volume of your blood. If you increase the volume of your blood, you can increase blood pressure. And so this is one of the ways in which the body can regulate and monitor and alter blood pressure within the body. And this is shown in this cartoon here. You have a pre-peptide, angiotensinogen, which is released by the liver. This is cleaved by another enzyme, another protease called renin, releasing angiotensin 1. Angiotensin converting enzyme then binds to angiotensin 1, cleaving it, carrying out a molecular reaction and producing a substrates which include angiotensin 2. This then acts on a variety of different parts of the body, including the blood supply, but also the tubular sections of the kidneys, targeting the nephron and increasing water retention. So if you're able to target and control the activity of angiotensin converting enzyme, actually you can modulate how much water is retained. And this is just a representation of what's actually happening at a molecular level. So here we have angiotensinogen. This is released by the liver. This is processed by renin, which is also a protease, which I've represented here by a pair of scissors. This binds to and cleaves angiotensinogen, releasing angiotensin 1. The peptide sequence of angiotensin 1 is shown here. What angiotensin converting enzyme does is it clips off one of the amino acids at the C-terminal of this peptide. By modifying the peptide, so it actually releases histidine and leucine, By removing those two amino acids, what it does is it produces the active form of the peptide, angiotensin 2. This then goes on and can bind to target receptors across the body, modulating physiology, and one of the products of which is the retention of water. This, of course, has a major implication for a number of human conditions. Increased blood pressure is associated with a number of really quite serious pathophysiological disorders in the human, increasing your risk of stroke, of heart attack, and so on and so forth. And so a huge amount of effort went into designing inhibitors that can bind to and inhibit the activity of ACE. And this drug, ramipril, does exactly that. What it does is it actually mimics the peptide structure at the heart of angiotensin 1. It binds to the active site as a competitive inhibitor, blocking the activity of angiotensin-converting enzyme. Important thing to note about this is that the more of the compound you have, the greater the inhibition, going back to the enzyme kinetics that we were talking about earlier. This is produced by Sanofi, and what it does is it decreases fluid retention, so basically allowing more water to exit the body during the elaboration of urine. By doing that, it reduces blood pressure, and this is one of the key drugs in the medical profession's armory to target high blood pressure. If you have high blood pressure, ramipril is one of the drugs that you will be prescribed. The final example of an enzyme inhibitor that I'll provide is targeting cyclooxygenase. Cyclooxygenase is responsible for the synthesis of prostaglandins. These are critical for inflammatory response within the body. And COX inhibitors can reduce the synthesis of prostaglandins by inhibiting the cyclooxygenase enzymes which are involved in the anabolic production of prostaglandins. This is the chemical structure of prostaglandin. And this is a summary of the anabolic processes that result in the production of prostaglandins. So you have a series of enzymatic reactions going from phospholipids to arachidonic acid and eventually to prostaglandins. And the key enzyme here are the COX-1 or COX-2 peroxidases, and these carry out a chemical modification of the phospholipids. Arachidonic acid rather, and convert it into prostaglandin. And actually this is one of the most common drugs that you could imagine. This is targeted by ibuprofen, which was originally produced by Boots back in the 1960s, 1970s. What ibuprofen does is it inhibits those cyclooxygenase enzymes. Again, it goes into their active site and competes against the substrate arachidonic acid, preventing the production of prostaglandins. If you have a fever, if you have inflammation in your limbs, then by taking ibuprofen, you block the activity of the cyclooxygenase enzymes, you reduce the production of prostaglandins, and that then dampens the inflammatory response that is going on inside your body, reducing your fever or reducing swelling due to inflammation in tissue. Now, this may be a good thing, it may be a bad thing, but it's one of the ways in which we can modulate a pathophysiological response to alter the biology of a medical reaction. So very briefly, what I'll talk about over the next few slides is a class of enzymes that are increasingly being targeted in human medicine. And these are protein kinases. And this is an article by Philip Cohen, now Sir Philip Cohen, who set up one of the key laboratories investigating protein kinases, established at the University of Dundee about 30 years ago now. And this is an article that he wrote back in 2002, talking about protein kinases as one of the key drug targets of the 21st century. So what are protein kinases? Basically, kinases are enzymes that form a key part of the signaling cascades that operate within all of your cells. Signaling cascades are one of the ways, or signal transduction pathways, are one of the ways in which your cells regulate the flow of information and control processes. These are the sort of command and control proteins within your cell that alter the metabolism of your cells, they alter the physiological role of your cells, they can control cell replication, they can control DNA replication, and so on and so forth. And they work by modifying other proteins with a phosphate group. Basically, they bind ATP and transfer the gamma phosphate from that ATP onto other molecules, frequently other proteins. By modifying those proteins, the phosphate group modifies the shape, the three-dimensional organization, the conformation of that protein, by altering its three-dimensional shape, you modify its function. And this is really one of the key conserved pathways through which life, in all its wonders, regulates the flow of information in cells. And this is represented in a very, very simple diagram here. So what you have is you have a protein substrate or a substrate of any kind. Kinases are also able to phosphorylate a large range of other chemicals within the body. You have a substrate and you have ATP. This is bound to the protein kinase, forming that enzyme-substrate complex that we were talking about earlier on in the lecture. And what the kinase does is it acts as a biological catalyst, breaking apart the gamma-phosphate bond shown here and taking this final phosphate from the ATP structure and basically attaching it to whatever the substrate is. As I say, in many cases, this is another protein. Obviously, phosphate groups are quite highly charged. That then alters the three-dimensional conformation of the protein and alters its function. The protein kinase then releases ADP and the substrate, the phosphorylated substrate. Now, increasingly, these are being targeted in the context of human disease, and in particular in the context of oncology. This is the crystal structure of BCR-ABL, which is a kinase domain, which is hyperactive in a number of different cancers, but in particular in some forms of leukemia. This is a substrate, a picture of the crystal structure of this kinase with an inhibitor bound in its active site. This is the inhibitor imatinib, and this is the chemical structure of that inhibitor. What this does is it inhibits the activity of this kinase. And what that does is it reduces the activity of the kinase. It acts as a competitive inhibitor and prevents this kinase overstimulating cells and causing those cells to replicate uncontrollably. Blocking that uncontrolled replication is one of the key ways in which we can target cancer, and increasingly there are a number of drugs which target cancers in this fashion. So just in the final 10 minutes or so, I'll talk about one of the other ways in which you can modulate an enzyme's activity. And this is by allosteric modulation. Up until now, all of the examples of inhibitors which are in clinical practice, remember all of the drugs that I've talked about are ones that are used in medicine to treat human disorders. So this final class, and the examples that I've given you are all direct inhibitors. They're either competitive or non-competitive inhibitors of enzymatic reactions. And in a way, that's the simplest way to target an enzyme. If it's got an active site, you can define the three-dimensional shape of that active site, and you can either screen for compounds that inhibit it, or actually design compounds which bind to that active site and prevent the enzyme functioning. However, in nature, one of the ways in which enzymes are modulated is by allosteric regulation. Basically, this is binding to a point away from the actual active site, but which has an impact on the shape of the active site. This, in turn, can alter the substrate binding or the active site function. And in many ways, phosphorylation is one way in which you can think about an allosteric regulation of a protein. If you chuck a phosphate group onto the side of a protein, it alters the shape of the protein. The active site is there. That active site will be altered in conformation, and the activity will be modulated. So what I've shown you in the earlier part of this lecture is a simple substrate product reaction plot for a simple enzymatic reaction. And this forms a very characteristic curve. Now, in the case of an allosteric regulation, where the enzyme activity is modulated by alterations in the three-dimensional shape of the protein beyond the active site, what you frequently observe is an S-shaped reaction, where the initial velocity of the reaction accelerates. It actually accelerates rather than shooting up immediately. You have a gradual acceleration and then the final topping out when it reaches Vmax. Probably the best studied example of this, or the best understood example of this, is actually one which is critical for human function, for the function of any organism which relies on oxygen. This is the binding of oxygen to hemoglobin. And here what occurs is that when you basically, in your blood, you have the complex formed by as hemoglobin, is actually made up of 4 individual globin subunits. When one of those subunits is bound to oxygen, this causes a conformational alteration in the whole complex, which increases the likelihood of oxygen binding to one of the other subunits. And so actually the kinetics of the reaction away from the initial binding of oxygen, that increases the likelihood of binding of oxygen, and that results in this S-shaped reaction in terms of oxygen binding. And actually, there are a number of ways in which you can modulate an enzyme through allosteric regulation. You can alter the activation. So here you have an active site. By binding an allosteric modulator of an enzymatic reaction, you can block an activation step and prevent a substrate ever binding. You can alter the formation of the active site by binding of an allosteric modulator, and here in the cartoon shape, you can see how the formation of that active site is altered by the binding of an inhibitor, preventing the substrate entering, but not actually binding directly to the active site. And a number of proteins, enzymes within the human proteome actually require binding to another protein in order to carry out their function. Hemoglobin is a very good example of this, where you actually have a tetrameric complex which binds oxygen within your blood cells. So if you have a protein which alters and blocks that dimerization, an inhibitor which alters and blocks that dimerization, then that in turn can inhibit the activity of the enzyme by preventing it ever forming an active complex. For a variety of reasons, these types of reaction are actually very, very difficult to design specific inhibitors for, mainly because it relies on binding to parts of a protein that are not the active site. An active site has a very, very well-defined three-dimensional structure which is designed to bind things. This means that in terms of drug discovery, you can design molecules which fit into that pocket, and you know what they're going to do. They're going to block the active site. That's relatively straightforward. For allosteric modulation, what you're often looking at are protein-protein interactions and very subtle modifications of the three-dimensional structure of that protein, binding to the outside of the protein and not to a well-formed cavity in that protein in the shape of an active site. Because of that, actually it's very difficult to design allosteric modulators which specifically target a given enzyme. What often happens is you have a large number of off-target effects, targeting other enzymes that you don't want to alter. Now, if you're exposing humans to those, what can result from that is actually a widespread disruption of cellular function because of that. And so for this and for a number of other reasons, it's actually very, very difficult to bring allosteric modulators targeting enzymes into clinical practice. There are examples of allosteric compounds which modulate, for example, the binding of agonists to receptors, so altering receptor activity. So that has entered into clinical practice. In terms of modulating enzyme activity, actually there are no clear examples where that is the case. So just to summarize, I've talked through what enzymes are, how you can modulate their function in a theoretical, conceptual way, and given you a number of examples of how we can do that in practice. In terms of background reading, I've posted a load of Nature Reviews and Drug Discovery articles on the internet. Don't feel you have to read those, but if you're interested in learning more, these will be very useful. And obviously, I'll be posting the screencast on YouTube, and we'll be sending the link through Blackboard. If anyone has any questions, feel free to come down to the front and ask them. If not, enjoy the rest of your afternoon.